Okay, class. So today we're going to do lecture 4, which is going to be a lot on the nucleus, genome structure, and chromatin. We're also going to cover DNA and DNA structure. And this is really where the course really starts to take off and everything starts to become very important. Today's lecture should be review for a lot of you guys, but I may throw out some stuff that you haven't been exposed to before, so really start to pay close attention. So as I'm sure you guys all know, DNA is the molecular blueprint of a cell. Everything that makes up a cell, it basically comes from the blueprint of the DNA, right? All of the different proteins and everything. Now, the bulk of DNA in a eukaryotic organism is stored in the nucleus, right? And the nuclear DNA is then further broken down into individual chromosomes. And most all of the genetic material is contained on those chromosomes. The one exception being mitochondrial DNA, which contains a whole bunch of different genes. So the chromosomes are actually composed of a protein, DNA, RNA complex, and that's called chromatin, right? It's not just DNA in the nucleus. There's a protein, DNA, RNA complex called chromatin. And the chromatin is a highly ordered structure that organizes the DNA in the nucleus, okay? And some of the things that are really important that we're going to cover in this course are DNA replication, DNA repair, transcription, and genome structure. And it's important to know that these are all tightly controlled processes, okay? And when you start to have problems with replication, repair, division, transcription, or genome structure, that can actually lead to diseases. So it's really important that we all understand how this works. Okay, so here's some more background. So as I'm sure you guys all know, genetic material is transferred, right? It can go from a parent cell to a daughter cell or from an organism to an offspring. In the case with DNA going from a parent cell to a daughter cell, that is basically called somatic transmission. And then when an organism gives it to its offspring, that's germline transmission. Here's a nice little immunofluorescence stain of a cell right here, and this is the nucleus, and here is the cell going through cell division, and you can see the condensed chromosomes going through mitosis. Okay, so as I'm sure you also know, genetic material contains genes, but maybe some of you guys don't know how that was discovered, right? So this was actually a discovery in the 1940s, right before sort of World War II. These scientists by the name of Beadle and Tatum did this experiment where they basically showed using metabolic mutations that it was likely that one gene encoded one enzyme. Okay, and this was really a big groundbreaking work, and they ended up winning the Nobel Prize for that idea that one gene leads to one enzyme. And they actually did it before they fully knew that DNA was genetic material, which just goes to show you how brilliant they were. Now, some of the things with genetic material, as I'm sure you guys know, it can be duplicated. And the segregation of chromosomes in a dividing cell is called mitosis. And we're going to cover the cell cycle in depth later on in the course, but figured I'd throw that out right now, especially because we have a dividing cell here undergoing mitosis. Some little tidbits that you guys might find interesting. So the human genome is approximately 3.2 billion base pairs. Okay, that's the size of it. It was sequenced the sequence was released in 2003, and it was actually started in 1990. Now, the sequence of the human genome is still not entirely complete, and it probably never will be, and you'll see later on in today's lecture why that is. There's a lot of repetitive elements, but ultimately it's close enough. We've pretty much identified most genes and know what, and now we're sort of figuring out what they all do, okay? So to take a good look at the cell, if you look at the nucleus right here, and you were sort of do a cross-section of it, all of the DNA is packaged into chromatin in the nucleus, okay? And it's this chromatin fiber here, which is composed of DNA, protein, and RNA, that basically allows a lot of the organization to occur in the cell. And it's very important that the nucleus be well organized, right? If you need to transcribe a specific gene, the cell needs to know where to find that gene. Now, the DNA and chromatin is all wrapped up in these histones, and we'll cover those at the second part of today's lecture. Here's a fully condensed chromosome over here, and just a few highlights that you guys need to know. You need to be able to identify the centromere and the telomere on a chromosome. Okay, and this is a fully condensed chromosome with basically 2 sister chromatids. Now, this is a really neat experiment. This is the experiment that basically proved that DNA was genetic material. And I've asked this as a question on exams, so you guys should sort of pay attention here. So the way this experiment was done, it was actually a 2-part experiment. So they had this strain of bacterium which caused pneumonia, okay, and it was smooth. And so the smooth strain of bacteria was pathogenic and caused pneumonia. They mutagenized it, and then they were able to pull out an R strain that was basically rough, and it was non-pathogenic. Okay, so now they have 2 different strains, an S strain and an R strain. And so they took this R strain and they mixed it with heat-killed S strain or cell extracts from the S strain, and this R strain became transformed. Okay, anytime you take up DNA, it's considered a transformation. Okay, and so this R strain became transformed, and it actually reverted back to, after it was transformed, it became very similar to the S strain that it was. And so basically the conclusion here was that there's molecules that can carry heritable information, and they're present in this S strain, right? So this S strain was pathogenic, this one was non-pathogenic. When you take the non-pathogenic one and mix it with an extract of this, some things can get transferred in a transformation, and restoring it back to the way it was sort of in the beginning. Okay, and then the second part was, well, what cellular constituent is that? Okay, and so they wanted to basically test whether or not it was RNA, protein, DNA, lipids, or carbohydrates. Okay, that's basically what a cell's made up of. And so what they did was they took their S strain, this is the pathogenic one, and they basically fractionated it into RNA, protein, DNA, lipids, and carbohydrates. And then they mixed it with the R strain, and then they tested which fraction caused the transformation back to the pathogenic strain, and it was the DNA fraction that did that. And so then the conclusion was the molecule that carries the heritable information is actually the DNA. And the R, when you mixed it with other things like RNA, proteins, lipid, and carbohydrates, the cells were not transformed. Okay, so I'm sure this slide's all review to you guys. I'm sure you've seen the structure of DNA, but let's just go over it again. So DNA is composed of nucleotides, and they're connected by a phosphodiester bond. DNA is actually a linear polymer, and it runs from 5' to 3' direction. And it's actually anti-parallel when it's with its complementary strand. Okay. DNA is made up of 4 different bases, adenine, cytosine, guanine, and thymine. And when it's assembled and it has this hydrogen bonding between the bases, it forms this double helix. Okay, and so you have your sugar phosphate backbone, and then the bases are in the middle, and they're actually hydrogen bonding. Now, this is Rosalind Franklin's X-ray diffraction data, okay, which led to the solving of the structure of DNA. And so the way X-ray diffraction works is she basically took some DNA and she crystallized it. And when you crystallize something, it becomes an ordered lattice, okay? And then she was able to put a beam of electrons through that crystal, and the charges basically on the DNA cause the electrons to diffract. okay, and then she put a piece of film in there and was able to see where the electrons were hitting as they were being bent. Okay, and so if you look at this, you can almost see here that you have your major groove here and also a minor groove that makes up the double helix. Okay, so this is basically how the bases in DNA base pair. It's complementary base pairing. They're connected by hydrogen bonds. Okay, so here's your sugar phosphate backbone on the outside, and you can see the DNA is running 5' to 3' and 5' to 3'. Now, adenine always base pairs with thymine, and guanine always base pairs with cytosine. And there's 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between guanine and cytosine. Okay, and just for completeness, whenever you have 2 rings, that base is considered a purine. So A and G, they both have 2 rings, and a single ring is considered a pyrimidine. So purines always base pair with pyrimidines. Here's a nice space-filling model of DNA as it sort of looks in the cell, and you can see here you have the minor groove and the major groove, and here is basically the structure of it. We'll get into this in detail in a second, but it's running 5' to 3', 5' to 3', and then you can see the bases pairing here. So here's sort of a blow up of that model. You can see the double helix structure, and it has this minor groove and this major groove that's present. And what's interesting, and we'll see this later when we're covering transcription factors, but most transcription factors, well, an alpha helix of a protein, which we covered in last lecture, actually fits perfectly in this major groove. And you'll see when we cover transcription that a lot of the transcription factors will have an alpha helix that'll sit down in here. So if you guys remember, we talked about zinc fingers transcription factors. Well, they have a little helix that can actually fit down right into this major groove here. Okay, and then they'll form non-covalent interactions with the DNA, and we'll cover that later in the term. Once again, here's the structure of DNA. It runs 5' to 3', and what links the sugars together is a phosphodiester bond. There's the phosphate. You can see the base pairing here between the bases, and the directionality of DNA from the 5' to 3' is basically where the phosphate is connected to the ribose. Okay, so if you look here on this ribose molecule, the base comes off of the C1 position. Okay, so that's carbon number 1. And so if you count the carbons, you can see carbon 1, this would be carbon 2. Right here we have carbon 3, where you have the phosphate attached to it, then carbon 4, and then carbon 5. Okay, so this is considered the 5' end, and this is considered the 3' end. And same on the other side here. So once again, DNA is actually anti-parallel. It runs 5' to 3' and 3' to 5'. Now, genes are encoded by DNA, and here's just a very, very rough schematic of sort of a way to think about it. You have gene A, gene B, and gene C, and they're encoded on the DNA. And when they're transcribed, you can get gene expression, and then the messenger RNA is then translated into protein A, protein B, protein C. It's usually not this simple. It's actually much more complicated. Genes can exist on the plus strand or the minus strand of DNA. Genes can have natural antisense transcripts, and there's non-coding RNAs and other things like that. So, you know, the genes can actually overlap. There's a lot of complications to it, but in its most simplest form, gene A gives rise to protein A, gene B gives rise to protein B, and gene C gives rise to protein C, right? The one gene, one enzyme hypothesis that Badel and Tatum had put forth. Now, as scientists, we look at DNA basically on computers mostly, right? And here's just a sampling of some DNA. DNA is actually read 5' to 3', and it was the microprocessor that really allowed us great advancements in understanding of DNA, right? Because we're now able to store huge amounts of sequencing data, and there's databases out there that have the entire human genome as well as hundreds of thousands of other different organisms. And the DNA sequence can be stored in many different formats, but two very popular ones are FASTA and GenBank. And so I've got a few questions here that I ask you, and I'm sure you guys have been exposed to this before, but do you know the difference between an exon and an intron? And in this image over here, the exons are actually highlighted, and they're the expressed portions, so it leads to what it basically leads to the protein. And then the introns are intervening regions, which actually get spliced out. And we'll cover splicing later on in the term, but I just wanted to throw those out to you right now. So it's important you know the difference between exons and introns. And we'll see the examples in just a second. Another thing that's really important is understanding the difference between the start of transcription and the start of translation. Okay, translation starts at the AUG codon, right? If it's read here, it's ATG, but when the mRNA is being translated, it's AUG, okay? The start of transcription usually occurs upstream of that, okay? And messenger RNA transcripts have what are called 5' UTRs on the ends of them, right? On the 5' end, there's a 5' UTR, and on the 3' end, there's a 3' UTR. And UTR stands for untranslated region. Okay, so the transcript is bigger than what is actually translated. And you guys will see this when we cover transcription and translation in upcoming lectures. Okay, so once again, DNA is replicated, okay? And when it's replicated, the individual strands serve as the templates, okay? So what ends up happening is the hydrogen bonds that hold the DNA together are actually broken, and then you have a template strand, and it basically reads the newly synthesized strand. Same thing, the other strand, the template strand S' here is read, and it becomes a new S strand. Okay, so this is considered semi-conservative replication. So this single piece of DNA becomes two pieces of DNA. And DNA is copied by an enzyme called DNA polymerase. I'm sure many of you guys have heard about Taq polymerase, right? Which is a Taq DNA polymerase from the organism Thermus aquaticus. And I'm sure many of you guys have copied DNA in one of your lab courses or in the lab at some point in time using Taq polymerase, okay? So this is what happens. It basically breaks the strand into two, and each strand is copied, and that's considered semi-conservative replication. Here's a nice schematic of what the nucleus looks like, okay? And here's a nice little microscopic image of a nucleus. And so some of the things that you guys need to know are the structures and the structural elements within the nucleus. Okay, this dense part that's stained right here, that's the nucleolus. Okay, and that's where a lot of the ribosomal RNA, as well as ribosomal DNA, are contained, and it's actually where ribosomes are assembled. Right, they're all assembled in the nucleolus. The other thing that you need to know is the nucleus is enclosed by the nuclear envelope. Okay, which is contiguous with the endoplasmic reticulum. There is an outer nuclear membrane and an inner nuclear membrane, and the way things get in and out of the nucleus is through the nuclear pore. Okay, now enclosing all of the chromatin within the nucleus are these intermediate filaments that sort of come together and create this little basket that sort of sits in here. It's sort of almost weaved together, and it's called the nuclear lamina, okay? And it sits sort of right on the outside along the inner nuclear membrane. Now, attached to the nuclear lamina or localized co-localized with the nuclear lamina is what's called peripheral heterochromatin. This is where the telomeres tend to hang out in the cell, as well as other heterochromatic DNA. And we'll learn the difference between euchromatic DNA and heterochromatic DNA later. Okay, but this sort of light blue shaded area right here is all sort of considered euchromatic DNA, and that's where basically most of the genes are, as well as other things. And then on the outside, you have your centromere and microtubules coming off, or your centrosome coming off, and with the microtubules coming off this centrosome. Okay. Now, what's interesting here, and I sort of mentioned this before, diseases can arise if there's issues with the structure of your genome, right? A genome structure is very, very important. And there's this disease called progeria, which is actually a mutation in nuclear lamina, one of the nuclear lamins, which forms the nuclear lamina. And what this mutation does is it makes it difficult for the peripheral heterochromatin, the telomeres and stuff like that to associate. You don't have the organization in the nucleus that you need. And what ends up happening is that people with progeria, it's a really devastating disease. It happens in children, and these children basically undergo rapid aging, and they have basically their telomeres get very short, and they just have a whole defect in their genome structure. And basically what ends up happening is little kids ages 12 to 13 die of old age, and have all of the symptoms of old age. So I encourage you guys to sort of look into that. It's sort of it's a really devastating disease, with somewhat interesting science behind it. So once again, here is the sort of schematic of the nucleus drawn, and once again you've got the nuclear lamina, the nuclear envelope, the nuclear pore, and here in light blue is euchromatin, and then in the dark blue you have heterochromatin. And just to make a note here, the difference between heterochromatin and euchromatin: heterochromatin is usually tightly compacted DNA versus euchromatin, which is sort of loosely packaged DNA that's quite accessible. So heterochromatin is largely inaccessible. Euchromatin is actually more open and accessible. All right, there's this neat technique that you can do, and it's called karyotyping, and this is actually karyotyping here. So if you take a cell and you basically block it during mitosis, where you have the fully condensed chromosomes, and then you squish it, you can actually get all of the chromosomes out in their fully condensed form. And then you can fluorescently label them with this technique called FISH, which is fluorescent in situ hybridization. And so this actually happens to be a human cell right here that they've sort of squished, and then they basically stained all of the different chromosomes, the fully condensed chromosomes, and then you can sort of arrange them. You can see here chromosome 1, chromosome 2, chromosome 3, and all of the different chromosomes. Okay, so this is considered karyotyping, and there's a process called karyogamy, which basically when you have two haploid cells that fuse together like a sperm and an egg, and their nuclei fuse, that's actually called karyogamy, when the nuclei come together, where you have two haploids and then it comes back together to become a diploid. Here's a nice schematic representation of the human genome. So here they are sort of color-coded. But if you do a schematic representation of the human chromosomes, you can sort of see them here. And once again, you have germ cells versus somatic cells. So a somatic cell will copy these, then divide, and so there's two copies of these, versus in germline transmission you end up with a haploid. And so a somatic cell contains two copies of each of these chromosomes, whereas a germ cell contains just one copy. Here you can see this little red line sort of marks the centromere on each of the chromosomes, and then on the ends of each chromosome are the telomeres. These little red dots here represent your ribosomal DNA. That's what gives rise to the ribosomal RNA, and these are all actually located in the nucleolus, right? They all come together to help form the nucleolus. And so I have a little question mark down here with trisomy 21. Here's chromosome 21. I'm sure you guys all know about trisomy 21. It's if you end up with 3 copies of chromosome 21, which can arise from incomplete mitosis or improper mitosis and improper meiosis during germ cell development, and a cell ends up with 3 copies of chromosome 21, it gives rise to Down syndrome. Now, there's actually other rare diseases where you can get a different copy, but why do you think that chromosome 21 is the one that's basically found the most in Down syndrome? Okay, and you don't find trisomy 3s and trisomy 2s. Well, the problem is that because chromosome 21 is so small, the cell can probably tolerate it. If you had too many copies of any of the other chromosomes, it's probably lethal, and during development the fetus would have self-abort. So one of the things that can happen with chromosomes is you can get chromosome translocations. Okay, so here's just a normal chromosome over here, and occasionally what you can get is a translocation where a part of one chromosome gets added to a different chromosome. Okay, and chromosome translocations are very common in genetic diseases where you get this sort of recombination, and we'll cover this when we're doing homologous recombination and just recombination in general. But one disease that comes out of a chromosome translocation is ataxia, and that happens when you get basically a translocation between chromosome 4 and chromosome 12. Chromosome translocations are also pretty common in cancer, and when we cover cancer at the end of the term, we'll actually look at some of the cells and all of the different translocations that can be found in cancers. But one really famous sort of translocation is what's called the Philadelphia chromosome. Okay, it's this tiny little chromosome over here. And what ends up happening is you have a chromosomal translocation where this end of chromosome 9 actually gets put onto chromosome 22, and this part of chromosome 22 actually gets added to chromosome 9. So now you have this 9 plus chromosome and this 22 minus chromosome. Okay, and this is what's considered the Philadelphia chromosome. And why this is so important is if this happens, and it happens fairly regularly in humans, you end up having leukemia called chronic myelogenous leukemia, or CML. It used to be one of the most deadly forms of leukemia that you could get, which was why this was actually identified. People wanted to know, because it creates this fusion protein. You have this gene ABL on chromosome 9 and this other gene BCR on chromosome 22. And when you get this translocation event, the BCR ABL, that makes a fusion protein called BCR ABL, which is a tyrosine kinase. And if this is around in the cell, it basically causes augmented signaling, which leads to disease. Interestingly, this is actually one of the most treatable cancers out there now, because there's this great drug called Gleevec, which is a tyrosine kinase inhibitor that's very specific to the BCR ABL fusion protein. Okay, so it used to be one of the most deadly cancers, and then someone discovered Gleevec, which inhibits the BCR ABL fusion protein. Okay, this is kind of an interesting slide. It's more for just your enjoyment than anything else, and it just goes to show that, you know, even organisms that are very similar, So here you have the Chinese muntjac and an Indian muntjac, basically coming from the exact same continent, separated only by a mountain range. They can actually diverge in sort of their chromosome structure, right? Here you can see all of the chromosomes in the Chinese muntjac versus the Indian muntjac that's right here. And even though these organisms look very similar, their genomes are, at least their chromosomes are very, very different, right? So the chromosomes can differ dramatically between organisms, even if they look somewhat similar or have the same sort of evolutionary ancestors. Okay, so here's also a very sort of simplified view of genes on a chromosome. This is actually a schematic of chromosome 3 from yeast, and you can see here that the orange colored boxes here are actually genes, and the gray is sort of intervening sequences. Okay, so you can see that genes can basically exist on the plus strand or the minus strand, and some of the things that you probably need to know, the yeast genome is very gene rich, right? And most of the genes are not spliced, versus the human genome, which is very gene poor, and almost every single gene is spliced. And if we look at the human chromosome, this happens to be human chromosome 22, and this is in its mitotic conformation. If we sort of blow up a little section of it, 10% of that chromosome arm will contain about 40 genes. These are sort of shaded here. And then if you take 1% of the chromosome there, you'll see that it contains about 4 genes. And then if you just look at one individual gene here, what you'll see is you'll have regulatory DNA sequences upstream, and then you have these exons and introns. There'll also be a 5' and a 3' UTR, and when you get gene expression, that will lead to a spliced mRNA transcript, which when translated will give rise to the protein, and then the protein will ultimately become folded. So here's just some statistics about the human genome. It's basically 3.2 times 10 to the 9th base pairs. There's a roughly 25,000 genes. The largest gene is 2.4 times 10 to the 6 nucleotides. The average is 27,000, and the smallest number of exons per gene. So you guys don't need to know this. It's just some fun statistics about the human genome. And if you sort of look at the human genome as a percentage and what it composed, the protein coding genes are actually a really small percent. It's this little red box shown at the end. That's the protein coding genes, right? That's the portion of the DNA that actually encodes for proteins. There's a large portion of DNA in our genomes that are just introns. These are actually transcribed, but then they're spliced out. We have sort of intergenic regions. These are sort of regions between genes that are non-repetitive, but they're neither introns nor codons, right, nor exons, right? So, and then you have these repetitive elements called line and sign elements and retroviral-like elements, okay? And these are all just repeated sequences that, as I mentioned before, they've sequenced the human genome, and they have a lot of this is sequenced, but they don't always have it all perfectly organized, right? Because these line elements and sign elements are difficult. Now, the vast majority of the human genome is actually expressed, and there's a lot of non-coding RNAs that have important regulatory roles in the genome. And we'll look at some of those as the course goes on. Okay. Now, here's just basically a nice little example of how it's believed chromosomes evolved. Here would be an ancestor chromosome with different portions, A, B, C, and D. And if you sort of looked at where those portions arise in a human, orangutan, and a lemur, and you did some fish down here, you can sort of see the different staining. So here's the A section stained in the human chromosome. Here's the A section stained in the orangutan, the blue, and here is the A section stained in the lemur. You can go through this yourself, but we'll just look at the C region. So here's the C region stained in the ancestor chromosome. Here it is in humans. And here's the C section in orangutan and the C in the lemur. So you can sort of follow those around. But it's basically, this can happen because chromosomes can invert and they can recombine and then revert again. You can have all these inversions and things like that happening to basically sort of rearrange the chromosomes themselves. And we'll look at some of the dynamic characteristics of the chromosomes when we get to sort of mitosis and meiosis. Okay, so we're also going to cover the cell cycle later in the term, but this is a great time to just like start to learn about it, because if you haven't already, there's two major phases in the cell cycle: interphase and M phase. And interphase is actually broken down up into three different phases: G1, S, and G2. S being the synthesis phase, and that's where the DNA is actually replicated. Okay, so you basically have the nuclear envelope surrounding your interphase chromosome, and then during S phase it's duplicated, and then during mitosis the chromosomes condense. They align along the metaphase plate. They're attached to the mitotic spindle, and then they actually get pulled apart in mitosis or M phase, and then get segregated into two individual cells that go back into interphase, typically G1 at this point. And so one of the things that happens here, which isn't shown in this thing, is the nuclear envelope. Actually, it does show it because the gray is the cell. The nuclear envelope actually breaks down during M phase, except in Saccharomyces cerevisiae. Here's a nice EM of DNA. You can see here the sort of the interphase DNA, and here's a sister chromatid fully condensed. So basically a fully condensed chromosome. And here is just some very important chromosome elements that I really want you guys to know. The chromosome, if you look at an interphase chromosome, you have your telomeres and your centromeres, then you have origin of replications, and then during interphase, when the DNA is being replicated, replication starts at the origins of replication, and you get these replication forks going off, and eventually the DNA is copied, and then the chromosomes condense, and they form these sister chromatids, and then you get basically the mitotic spindle attached to the centromere, and then those get pulled apart during mitosis, and then you're left with your interphase duplicated chromosomes that are separated in the cells. Okay, so that's it for part A of this lecture. I'll start part B now.